bacteria, virus, fungus, algae, and protozoa. These organisms do not possess specialized tissues and organs, as found in higher plants and animals. In general, all living cells are broadly classified into two protist, prokaryotes, and eukaryotes. The bacteria are under prokaryotes. And the fungi, algae, protozoa, plant cells, and animal cells are classified under eukaryotes. In prokaryotes, the word pro denotes before. Karyon denotes nucleus. That means they lack proper nucleus. In eukaryotes, the word eu denotes true. Karyon denotes nucleus. That means they have proper nucleus. So, in microorganisms, the bacteria is classified under prokaryotes. The fungi, protozoa, algae is classified under eukaryotes. But the virus, they are neither under prokaryotes nor under eukaryotes. Because the virus does not carry any organelles or other specialized structures. Virus structure is different from others, which will be detailed in separate video. The difference between prokaryotes and eukaryotes are: size-wise, eukaryotic cells are 10 to 100 times larger than prokaryotic cells. Prokaryotic cells do not contain nucleus. Instead, they have nucleus-like material, that is, nucleoid, which lack a nuclear membrane. But, eukaryotic cells possess well-defined nucleus in it. Prokaryotes are unicellular, whereas eukaryotes are unicellular and multicellular. Cell wall of bacteria is rich in peptidoglycans. On contrast, eukaryotic cells like animal cells lack cell wall. But, plant cell possess cell wall, which is rich in cellulose. And fungal cell possess cell wall, which is rich in chitin. Ribosome size. Bacterial cells possess smaller ribosome on comparison to eukaryotic ribosome. That is, 70s ribosome in bacteria, and 80s ribosome in eukaryotic cells. Mitochondria, powerhouse of cell, absent in prokaryotes, but present in eukaryotes. Endoplasmic reticulum, absent in prokaryotes, but present in eukaryotes. Golgi apparatus, absent in prokaryotes, but present in eukaryotes. So, membrane-bound organelles like mitochondria, endoplasmic reticulum, Golgi apparatus, are present in eukaryotic cells. But they are absent in bacterial cells. Another membrane-bound organelle, chlorophyll, present only in plants. Generally it is absent in fungi and bacteria. But exception, present in certain bacteria like cyanobacteria. Cytoplasmic membrane. In prokaryotes, cytoplasmic membrane do not contain sterol. But, in eukaryotes, cytoplasmic membrane contain sterol. These are the difference between prokaryotes and eukaryotes. So, the eukaryotic cells possess well-organized nucleus, and membrane-bound organelles like mitochondria, endoplasmic reticulum, Golgi apparatus in their cytoplasm. But, the bacterial cells does not possess nucleus. Instead, they have nucleus-like material, that is, nucleoid. And they do not have membrane-bound organelles. The detail about the structure of prokaryotic cells will be discussed in separate video. Schematic illustration of structure of bacterial cell is available as downloadable link in the below YouTube description. Coming to the introduction to mycology. Mycology. Study of fungi. The Italian botanist, Pier Antonio Michel, who is popularly well known as the father of mycology. Pier Michel is renowned for his research in plants and fungi. The disease caused by fungi is termed as mycoses. What is fungi? Fungus is an eukaryotic cell, spore-bearing, or spore-producing organism, which lack in chlorophyll. So, they cannot make their own organic food. And they are chemoheterotrophic organism. That is, they uses chemical as source of energy, and organic compounds as source of carbon. Based on the nutritional type, organisms can be photoautotroph, which uses light as source of energy, and carbon dioxide as source of carbon. Photoheterotroph, which uses light as source of energy, and organic compounds as source of carbon. Chemoautotroph, which uses chemical as source of energy, and carbon dioxide as source of carbon. chemoheterotroph, which uses chemical as source of energy, and organic compounds as source of carbon. So the fungus is a chemoheterotrophic organism, which uses chemical as source of energy, and organic compounds as source of carbon. Morphology of fungi. They are unicellular and multicellular. Example, yeast, which is a unicellular organism, and mold, which is a multicellular organism. Yeast, they are microscopic, requires microscope to visualize. But molds are easily seen with unaided eyes. In this video, you can observe the growth of mold. This is a fast-forwarded video. Fungal contents are protected and encased inside the protective barriers like cell wall, followed by cytoplasmic membrane or plasma membrane. These structures protect the fungus from many adverse conditions. So, there are antifungal agents, which is used to target against these structures. Cell wall. They are rich in chitin and glucan. They play an important antigenic determinant for the fungus. In animal cell, there is no cell wall. In plant cell, cellulose is the major cell wall component. And in bacterial cell, peptidoglycan is the major cell wall component. Cytoplasmic membrane, or plasma membrane. Unlike bacterial cells, these membrane contains ergosterol and zymosterol. Next. Nutrition. Fungi are chemoheterotrophs. Lack chlorophyll. Embedded in food source or medium. And undergoes external digestion. They are chemoheterotrophic organism. Uses chemical as source of energy, and organic compounds as source of carbon. They lack chlorophyll. So they cannot make their organic food. They are generally embedded in food source or medium, like wood, vegetation, leather, bread, and so forth. They undergo external digestion. That is, fungal cell secretes extracellular enzymes. These secreted enzymes digest the outside food material. So the digestion is completed outside the organism, and then, these digested end products are absorbed by the fungus. This is an external digestion, which is seen in fungus. Fungal reproduction. Unlike other organisms, both asexual reproduction and sexual reproduction is seen in fungus. Asexual reproduction occurs by budding or fission. In budding, progeny cells buds off from the parent cell. In fission, a fissure is created in the middle, and then the two cells separates. This thing happens by mitosis. That is, from single cell, two cells are formed. As a result of asexual reproduction, asexual spores are produced. Examples of asexual spores are arthrospores, blastospores, chlamydospores, cunidiospores, and sporangiospores. Next. Sexual reproduction occurs by fusion, or union of gametes. That is, fusion of male and female gametes. This thing happens by meiosis. That is, from two cells, one cell is formed. As a result of sexual reproduction, sexual spores are produced. Examples of sexual spores are zygospores, ascospore, basidiospore, and zoospores. How fungus are classified, especially based on their morphology. Number 1, yeast. 2, yeast-like. Next, mold, or filamentous fungi. And lastly, dimorphic fungi. First we will see about yeast. There are single-celled organisms, that is, they are unicellular structures. Microscopically, their size is larger than the bacteria. There are well-grown at 37 degrees Celsius, which is the body temperature. Next. Microscopically, they are oval to round-shaped cells. The picture shown here is the actual microscopic image of the yeast cells, which appears oval to round shape. Macroscopically, after incubation, they appears smooth surface colonies, which resembles like bacterial colony. Reproduction. Here, the progeny cell buds off from the parent cell, and the process is termed as budding, which is an asexual reproduction. In summary, yeast is a unicellular organism. They are oval to round-shaped cell size, larger than the bacteria. They are well grown at 37 degrees Celsius. After incubation, it resembles like bacterial colony, that is, smooth surface colonies. Reproduction happens by the asexual process called budding. Cryptococcus neoformans, one of the example for the pathogenic yeast. Next, yeast-like fungi. This organism also, similar to yeast. But, they give rise to an elongated structure, which is termed as pseudohyphae. Candida albicans, one of the example for the pathogenic yeast-like fungi. In summary, yeast-like is also an unicellular organism. They are oval to round-shaped cell. But, they give rise to pseudohyphae. Like yeast, they also well grown at 37 degrees Celsius. After incubation, it also resembles like bacterial colony, that is, smooth surface colonies. Reproduction happens by the asexual process called budding. All the features are similar to yeast, but they possess pseudohyphae. Next, mold, or filamentous fungi. Sometimes it is also spelled as mold. Both are correct. They are multicellular structures. They give rise to the filamentous structures, which is termed as hyphae. They are well grown at 25 degrees Celsius, that is a room temperature. After incubation, cottony and spreading type of growth will be observed. Reproduction happens by both sexual and asexual process. This hyphae are two types: septate hyphae and aseptate hyphae. In septate hyphae, the hyphae have cross walls in between. But, in aseptate hyphae, the hyphae do not have cross walls in between. These aseptate hyphae, also termed as cenocytic hyphae. As we already discussed, the mold give rise to the filamentous structures called hyphae. These hyphaes branches in all direction and form a mat-like structure, which is termed as mycelium. So the mycelium is nothing but the complex form of hyphaes. In mold, the term thallus means fungal body, which constitutes of mycelium and spores. Molds are something like a microscopic version of plants and trees. Their structures are similar to plants and trees. They also have root-like structure, which is essential for the absorption of nutrients. Also have structures like stems and branches, which give rise to the fruiting bodies, like spores, in mold. So, in mold, the mycelium, which is exposed out, is termed as aerial mycelium, which is also called as reproductive hyphae, because they are involved in the reproduction and produces spores. The mycelium, which is embedded inside the surface, is termed as surface mycelium, which is also called as vegetative hyphae, because they are involved in the attachment to the surface, and also for the absorption of nutrients. This is a fast-forwarded video. In this video, you can observe the mold growth, which is cottony and spreading type of growth. As we already discussed, this is an aerial mycelium, which is exposed out, which is also called as reproductive hyphae, because they are involved in the reproduction and produces spores. Now you can observe the cottony and spreading type of mold growth. This is the mycelial growth under microscopy. These are all fast-forwarded videos showing the mold and their growth. In summary, molds are multicellular structure. They give rise to filamentous structures called hyphae. These hyphae form a mat-like structure called mycelium. There are well-grown at 25 degrees Celsius, that is, room temperature. After incubation, it forms cottony and spreading type of growth. Reproduction happens by both sexual and asexual process. Dermatophytes like Microsporum, Epidermophyton, Trichophyton, are the examples for the mold. In addition, some molds have root-like part, arise from the hyphae, which is termed as rhizoid. This structure anchors the fungus to the surface. Rhizopus, one of the fungi, possesses this structure. You might be knowing, this type of fungus-infected bread, which is caused by Rhizopus stolonifer, which is also well known as bread mold, also possess rhizoid for anchoring. Lastly, dimorphic fungi. The word dimorphism term is used to describe a fungus which occurs in two different forms, depends upon environmental conditions, like temperature or place. Depending upon the environmental conditions, this type of fungus exists as yeast and mold. At 37 degrees Celsius, that is, body temperature, it grows as yeast. At 25 degrees Celsius, that is, room temperature, it grows as mold. Coccidioides immitis, Paracoccidioides brasiliensis, Blastomyces dermatitides, Histoplasma capsulatum, and Sporothrix shanaki, are the four pathogenic dimorphic fungi, which exist as both yeast and mold, depending upon the temperature. With this we are coming to the end of Part 1 lecture on Introduction and Classification of Fungus. In next video presentation, in Part 2, we will discuss in